So our first speaker is Sarvagya Saluja, a postgraduate associate in the Department of Molecular, Cellular, and Developmental Biology. Sarvagya is currently interested in how to make glue from the cholera bacteria that can be used in a wet environment. A fun fact is that Sarvagya won 2 dance battles with a broken foot last year. Pretty fun.
All right, so today I will be talking to you about venoms and antivenoms, and yes, I am Sarvagya. All right, so before we get started, I was told to tell you a little bit about myself. Well, hello? This is not... Perfect. All right. So I was told to tell you a little bit about myself and my journey into science, why I'm here. So this is me. But when I was young, my mom used to bring home a lot of books on nature and animals, and I remember reading about some really cool creatures, like a fish that looks like a rock. Oops, a rock, but if you step on it, it'll kill you. And a fish over here, this dude, that apparently is yummy enough to die for, because it's a delicacy in Japan, but it still kills over 100 people every year who try to eat it. So I was born in India, and then when I was 1, I moved to Mexico. That's where I grew up, and then when I was 18, I went to college at the University of Minnesota. Here I studied material science, so basically just the properties of materials, like things like plastics and metals and what you can do with them. And there I also took a really interesting neuroscience course, so I studied the brain a little bit, and that brought me to biology, which is what I'm doing here at Yale. So I work on cholera. All right, so let me get started with what I'm really here for, so the juicy stuff. So I'm sure a lot of you have heard of at least a few animals you should be wary of. So spiders, spiders, don't touch them. They'll sting you. Snakes, don't touch, they'll bite you. Tigers, definitely don't touch them. They'll eat you. So do not touch. There's millions of animals here in the world. Why are some of them especially dangerous? So, yes? Some, yes. Some of them have venom. Some of them have big teeth, like tigers. Some of them are really fast, like cheetahs, and they can use all these techniques to effectively feed themselves and defend themselves against predators. So all of us in this room and these animals all have found effective ways of doing so and keeping their species alive. And some of them, like spiders and some species of jellyfish, have found something called venom, as you said. So what exactly is venom? Venom is basically an injectable poison that's made of harmful toxins. So toxins are little chemicals that are toxic for your body, as the name suggests. And it's venom when it's actually injected into your body through either fangs or maybe little stingers, like this jellyfish that has nematocytes, which it uses to inject the venom into your body. So, all right, there's millions of animals, and they all have so many different types of venom. How do we start to understand them? So we can start to classify them by what they do to our bodies, So some creatures like the cobra, the brown recluse, Russell's viper, and pseudoscorpion have toxins in their venom that are actually able to start eating away at the flesh around the wound or the puncture bite. And this causes things like swelling, a lot of pain. So literally your skin is being eaten away, very painful. And if you don't treat it on time, you can lose a lot of flesh or even whole limbs, your whole limb. So you can have it amputated. Some animals contain something called a hemotoxin in their venom. So animals like the rattlesnake or the pit viper are able to inject chemicals into your body that actually start breaking down the blood vessels that carry the blood throughout your body. And it causes a lot of internal bleeding, so hemorrhages. And so this bleeding is really terrible, but sometimes you can bleed so much and causes your blood pressure to drop, which can actually give you a heart attack, so you die even faster. Really, terrible stuff. Finally, some animals contain something called a neurotoxin in their venom. So the most dangerous animals by far in the world, venom-wise, contain neurotoxins in their venom. So animals like the blue-ringed octopus, the black mamba, stonefish, and the box jellyfish contain toxins that are able to stop your brain from communicating with the rest of your body. And this can cause paralysis or cause your lungs to stop pumping air in and out. So respiratory failure. If you don't get air, you die. So I talked about cytotoxins, hemotoxins, and neurotoxins. Unfortunately for us, animals don't usually have just one kind of toxin in their venom. They often have 2 or even 3, they can have many, 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 but the effects can be all of the above. So your skin can start literally being eaten away. You're bleeding. You can't move. So everything can start happening at once. Really awful things. So for this talk, because I think it's really interesting and it's found in the most dangerous animals, I'll be focusing on neurotoxins. So neurotoxins are the deadliest. Why? So for this, we need to understand what the brain does. The brain is our body's command center, right? It's able to take in inputs from the rest of the body, like the eyes and our skin, like the temperature, and tell the rest of your body what to do. So if I'm walking this way and I'm like, whoop, I don't want to crash into this table, my brain is able to see the table and tell my legs to stop moving. So since the toxin attacks the connection, it disrupts the connection between the brain and the cells in our body, it can stop all of this from happening. So even the things that we don't realize that the brain is controlling, like it keeps our digestion happening and our lungs moving. This is the inland taipan. It's the most venomous snake in the world. And each of its bites could kill 100 people. And just to make sure, when it bites you multiple times. So, yeah. So how exactly does the brain talk to the cells in our body? It does this by using nerve cells or neurons. So this guy carries the signal that the brain is sending all the way down to the cells, say in your muscle. And then at the very end, the muscle sends out chemicals called neurotransmitters. These guys that go and attach to your muscle cell, and they communicate what they want you to do, right? So the toxins they're traveling in your body, and they attach to your muscle cells instead. And that way, the chemicals that your brain is sending out aren't actually able to reach your cells anymore. They're blocked. So in this little drawing, so the cell neurons are saying, please move, and you can hear it at first, but then the muscle cell doesn't know what's going on once the toxins have entered your system. So they're blocked. So you must be thinking, is there any way we can stop all these horrible, terrible things from happening to us? The answer is yes. So our body has a natural defense system in place, our immune system, which some of you might have heard of because of COVID. We really needed our immune system to step it up and protect us against germs and viruses. virus in the case of COVID. So there are multiple levels of defense that we have. So the first thing is like physical barriers, like our skin and mucus, our nose hairs. They stop things like from physically entering our bodies. What happens if something makes it in regardless? So maybe you inhale it. Then our white blood cells kick in, our innate immune system, and they're basically able to recognize when things should not be in our bodies and start like basically a generic response to get rid of them. So things like fevers are your body warming up to kill the bacteria or the virus, neutralize it, and get rid of it from your body. But a lot of germs and toxins are able to bypass the level 2, the second line of defense of your immune system. And what you need is more specialized responses. So this is when the third level comes in. So your body is able to create little weapons, like specialized weapons that know exactly what's going on with this toxin and how to deal with it the best way possible. So these are called antibodies. Antibodies are specific to what toxin is going to enter your system. How do they do it? These antibodies come, and as I showed, like they go and they stick onto the toxin. They attach to it really hard, and basically the toxin can't do what it was originally doing in your body anymore. So in this case, if the toxin was plugging my little muscle cells' ears, it can't do it anymore because the shape is different. And another thing it does is it makes it easy for your cells to recognize that they should be gotten, these toxins, like now we can get rid of them. So basically you can just poop these out. Now you turn something harmful into something harmless when the antibody is stuck onto it, and you can just get rid of it. This is a cute little comic that I found that says like, I'm hit, go on without me. And it's like the bacteria trying to avoid the immune system. So it's little cells that send out the antibodies that go and stick onto the bacteria. This could also be the toxin in the venom. These antibodies are the essential ingredient to antivenom. So how exactly do we make antivenom? What does it do? So first, we extract the venom from whatever venomous creature we want to make the antibody for. And this guy right here, he's milking it. He's milking the snake. And then we inject it, small amounts, very small ones, into an animal that is able to create antibodies. So its body reacts to the venom that we're injecting, to the toxins, and starts circulating antibodies throughout the blood. And then what we do is we take the blood, we take those antibodies. We don't kill the horse. But we take those antibodies and we use their antibodies to help ours. When venom is injected into our system, yes, our immune system starts defending our bodies. But the thing is, there's so much toxin in our bodies that the antibodies can't keep up. We don't have enough to combat the toxin, the amount of toxin. So we need help. We picked a fight, and now the horse's antibodies are our backup. All right, so if we can make antibodies, why do hundreds of thousands of people still die every year from envenoming? This is true. This is a map of people that die every year from snake bites only. And as you can see, so the darker areas show places where more people die from snake bites, and the lighter areas show where less people die from snake bites, so less than 10 in areas like the US and Australia, even though Australia has some of the most venomous creatures in the world, and more in like areas in Africa and some areas in South Asia like India. What's going on? So just like us, more venomous creatures exist in warmer areas. So they're able to stay alive, so there's a much higher density of venomous species right around here, right around the equator. And basically, there's also more people, so there are more encounters with venomous creatures. So more encounters means more people are likely to die before they make it to the hospital. Yes, that's another thing. It's very important that when you're bitten by something, like time is of the essence. You need to make it in 30 minutes to maybe an hour or 2 hours to the hospital. Otherwise, the venom will have caused so much damage to your body that there's no going back. So if you live really far away from the hospital, so people in rural areas in some of these communities, they aren't able to make it to the hospital on time. That's it. And finally, venom is really expensive to make. The whole process piles up, and just one person needing to go to the hospital, that can cost up to $30,000. So a lot of these communities, the hospitals either don't have the money to have this antivenom on hand, or people just can't pay for it. So they can't manufacture it, they can't pay for it. So this is a distribution problem. Who deals with it? So people in public health are in charge of figuring stuff like this out. So some of the initiatives in public health to combat this problem is, for example, this guy, Tukaram Rao, he started handing out boots in his village in India. So there wasn't enough money for antivenom, and thousands of people were dying. So basically, he started handing out boots, and the fangs couldn't penetrate the rubber in the boot. So instead of trying to figure out how to fix the problem after someone got bitten, this is an initiative to prevent it from happening. And in Nepal, I thought this was super interesting. There's a motorcycle volunteer program. So for people who don't have transportation, they can't get to the hospital quickly, they started this program where people on motorcycles, they told them exactly how to get to the closest hospital that had antivenom. And basically, these people, even if they didn't know where to go or how to get there, they knew to find the closest guy with a motorcycle and just get on their motorcycle and go to the hospital. And the people with the motorcycle were compensated for their gas money. So this took the death rate from 10.5% of the people who were bitten to 0.5%. So it decreased it immensely. Very successful. And what does this mean? It means that in areas where maybe there isn't that much money, other things can be really effective. So aggressive community responses, the community coming together, is really important. And there isn't just one solution to our problems. So with that, let me summarize the stuff we talked about today. So I talked about what venom is and why it exists. So basically, venom is an injectable poisonous substance developed by animals to effectively defend themselves against predators and find prey, so feed themselves. I talked about the types and effects of venom. So I talked about cytotoxins. They start eating away at your flesh. Hemotoxins that break down the blood vessels and cause bleeding, and neurotoxins that impact the connection between your brain and your body. I talked about how our body naturally has systems in place to defend ourselves against things like this, our immune system, and creates specialized tools against each different foreign invader that it encounters called antibodies. And what the antibodies do is they go and stick onto this invader, and they stop it from being harmful. They make something harmful into something harmless. And you can just get rid of that. After that, I talked about how we make antivenom. We can take small amounts of venom from the dangerous creature and inject it into another animal that's good at making antibodies and use that to make antivenom. Finally, I talked about what's going on today, so public health-wise, and initiatives people have in place to combat this crisis of envenoming. Thank you so much. I hope you learned something, and I'm happy to take any questions. Yes. Feel free to raise your hand if you have a question. Next question. Yeah.
How do you doesn't you if you don't have an antibody, doesn't you have a cell that gets a part of an invader and then sends it to another cell, and then they make more antibodies of the thing?
So do you mean if you don't have antibodies, your body is able to start making them when it realizes something's wrong? Yeah, that's true. So your body does start making antibodies when it realizes that there's something harmful in your body. That's a lot of bodies. That is what helps us when we're sick. So it goes and sticks onto the viruses and bacteria and helps us get rid of them. Makes them harmless and helps us get rid of them.
Are any other animals, other than horses, immune to snake venoms?
So horses, they don't start out immune to snake venom. When we start injecting them with small doses of venom, we can start their production of antibodies. So their immune system reacts just like ours and starts producing these antibodies. And then basically we can increase the dose to the point where there's enough that we can extract some of it and it's useful for us. And that way we can make a lot of animals produce the same antibody, so it doesn't have to be a horse. But horses are big, so they produce a lot of antibodies, and they're also really nice, so they're easy to hang around. So you could try with an elephant, I guess, but they're kind of hard to come across and handle. So most people use just an in-between. So you can use a mouse too, but then you'd have to have so many mice. Yeah. Yes?
If rattlesnakes bite you, does it stay in the same spot where the rattlesnakes bite you, or does it spread around?
The venom?
Yeah.
The venom, so it does start spreading around. That's why one of the things that they tell you is to lay as still as you can after you've been bitten. So contrary to what a lot of people think, most of the venom doesn't actually spread through our bloodstream. So that's already circulating. It's our immune system has another set of tubes in place, and that circulate something called lymph fluid that carries all the toxins and venoms. And it doesn't move usually too much if you're not actively moving. So try to stay as still as possible because it does start circulating throughout your body. And eventually it will, but you can slow it down. Yes. Yes?
Yeah, so we use horses as an example. So the horse body is somewhat different from the human body. How do we know that, the horse body, for all I know, they have some unique attribute about them which makes them produce a different kind of antibody? Yes. How do we know that certain animals will make the proper antibodies that will correspond to humans and work the same way?
That's a really good question. So you're right. Horses are pretty similar to us. They're mammals and they're large and they have their blood circulating. A lot of things are similar to us, but a lot of things, some things are not. And you're right, they do create antibodies that aren't exactly perfectly matched with the ones that we would need. So antivenom can help, but it's not perfect. So the farther away the creature that we're using is from how our bodies work and the antibodies we produce, the less effective the antivenom will be. So it's good, it's pretty good, but it's not perfect. So yeah, you're right.
Okay, we'll take one or two more questions. Now, let's give the speaker a round of applause. Thank you.
All right, so moving on to our second speaker. Our second speaker is Cynthia Lowe, a third-year MD PhD student in the Yale School of Medicine and the neuroscience program here at Yale. Cynthia is interested in studying how neurons, blood vessels, and tissue structure outside the cell work together. A fun fact is that Cynthia became interested in neuroscience after mistaking a book in her middle school library for a physics book, since it was called Newton's Madness. And so Newton is a famous physicist, but it was actually about patients with neurological disorders. So that was a happy accident, I'm sure, that led Cynthia to be interested in neuroscience. So sometimes accidents can produce pretty cool effects. All right, we can get started.
So, I know you all came here today for the Science Café, and I'm just going to add a little bit of a modifier called material science. But I heard that I'm supposed to talk a little bit about myself, so we're going to be talking about material science, which admittedly when I was your age, I had no idea it even existed. So actually when I was a little kid, I was actually interested in three somewhat different things. I was interested in being a scientist. I thought medicine was really cool because of that Newton's Madness stuff, and I also thought that engineering was pretty cool because the whole thought of building stuff was also pretty cool. And so at this point in my life, as an MD PhD here at Yale, I'm kind of training to be a combination of all three due to kind of how I've gotten here. And so what exactly does it mean to be a physician scientist in training? And so for me, what it means is that I do a combination of a lot of things in my work, and I'm also interested in things that are at the intersection of a lot of different topics. So I had to study a lot of biology, a lot of physics, a lot of chemistry, particularly when I was in your age. I was interested in all those things, and I still continue to study that now. When I was in college, I did a lot of studying of materials, do a lot of making and a lot of testing of materials. And here at Yale, I've been very interested in neurology, and I've honestly been kind of reading about it since I was your age. But now I get to actually see patients with neurological conditions along with all the other types of patients. And so I went to college at MIT. I majored in material science. Then I worked in 3D printing for 2 years. And then I came here, and my current lab is actually an bioengineering lab. But we work on blood vessels, and we also work with other clinicians on studying their conditions. So I'm going to be studying a condition called epilepsy, which causes these things called seizures, which affect the brain. So, as I say that, seeing as I'm a materials kind of girl, I would like to invite you into my materials world. So some souvenirs to look out for as you traverse through my materials world. I would like you to find out what makes a material scientist different from other scientists. What are some different ways to describe materials, and how do we change materials to make them different? Because that's kind of what we do. So, if you're like me, and someone told you, you should become a material scientist in college, and you're like, I don't know what that is, you go to Google. And what does Google say? Material science is an interdisciplinary field concerned with the understanding and application of properties of matter, connections between underlying structure of material properties, process. What does that even mean? And this is why no one knows what material science is. Some people might say, it sounds like chemistry, right? Matter, chemistry, stuff. It's not quite. And so one way to think about it is that chemistry focuses on what something is made out of, and material science likes to think a lot about how something is made, because that can be just as important as what something is. So let me give you one example. Should you ask a chemist or a material scientist for advice in picking your jewelry? So let's say you want to go shopping for an engagement ring. Your engagement ring, you want it to be made out of carbon. Here's your options: a piece of coal, some graphite, and a diamond. The chemist says, what's the difference? It's all carbon to me. And the material scientist says they may all be carbon, but they have different structures and they are formed differently. They are entirely different materials. And if you take a look at it, they do in fact have very different structures, and this is what makes them different. Even though they're all made out of the same atom, so that little black ball thing, which is what we call carbon, because we arrange them in different configurations, you get something that is very, very different. Because I guarantee you, if you give someone a piece of coal and you tell them that is going to be their engagement ring, they will probably get a little angry at you. So pick the diamond, go with the material scientist. And so just kind of to sum up with material science, this is what they call the material science tetrahedron thing. But basically what it is that we look at a combination of things. Structure is what I told you, how you arrange stuff is very important. Properties is what does that arrangement of stuff give you? Does it make it hard like a diamond? Does it make it flammable like coal so I can use it to heat up my house? Performance is how well does it do that, right? So does your coal give you like a little bit of flame, or does it make you really, really hot? Processing is what did you do to make it like that? And characterization is the testing you do to make sure that it actually does what you say it did, because that's very important. And so if you ask, like, what exactly does all this cover? The answer is everything. Engineering materials covers things like metals and alloys, so all the gizmos and gadgets and cars and things you can think of. Ceramics, you might think of bowls and plates and stuff, but I guarantee you ceramics is actually a lot more important than that. Bricks are actually made of ceramics. A lot of things that you need to be hard and resistant to a lot of heat are made out of ceramics. Polymers, plastics, anything made out of plastic, which I guarantee you you're probably either wearing, holding, or you have on your body right now, is made out of plastics. Composites, which is just a mixture of stuff, is very important. Big field of research right now is, for instance, making stuff that is lightweight for things like planes and stuff. Semiconductors, you all have phones, yes. So technology, you need semiconductors, and biomaterials, which is where I'm kind of in. So that's kind of in the field of looking at nature and how nature makes materials, and then how can we copy that or make something like it to help us make, for instance, tissues or other things of that nature that's important and useful for us. So I promised you that we would go to a cafe. And now, I think you all picked the diamond, right? We were going ring shopping and you all picked the diamond. That means that you are hired. It is time to start baking. Welcome to the kitchen. And so today we're going to be making bread. Bread is a very good example of material science. Why? Because there are a lot of different types of bread. But you know what? They are made out of the same basic ingredients. Flour, water, yeast or baking soda, salt. Some people use eggs, some people use milk. That's kind of up to you. But ultimately speaking, you kind of just use the same basic set of stuff and you get some very, very, very different results, right? In fact, you can open up a whole bakery just based off of the same basic ingredients, right? And that's kind of material science in a nutshell. And so the question is, what do all these different things do for us? So one important part is what we call gluting. Flour has these components that when we combine it together, they form proteins that then create basically like a bit of a net-like structure that makes our network that we'll see later kind of gives the bread the chewiness that we think of. Starches. So in order to inflate this network and make our bread something a little more than just a flat pancake, we actually need air inside our bread. So starches feed the yeast that are our rising agents. So either yeast or baking soda or things like that. We need gas in order to make what we call pores, but basically to rise our bread. And we need heat and other agents like eggs and things like that to basically make sure everything stays in place, because you would not want to hand your customer a piece of bread and have it deflate on them. And so let's start our bread. So what are the first things we do? You put everything into a bowl and then you start kneading. Yeah? I mean, there's some no-knead recipes, but that's a different thing. So when you put your flour and your water together, the proteins in the flour basically come together, and they form what is called a gluten network. And this is what gives us the property of the stretchiness of our dough, right? If you've ever picked up pizza dough or anything like that, it's nice and stretchy the more and more you knead it. And this gives us what we call, the performance of our bread, which is what we are looking for. And what we want, we don't really care about how stretchy the dough is at the end, right? What we want is a nice, soft, and chewy bread, right? And so that's the difference between property and performance. And so as you knead the bread, you make the network short, long, short, long, short, long. And then as you squish these things together, you make this network stronger and stronger. And one example, for instance, of why networks are important, actually, it's not just in bread, but also in glass. And so this is, glass blowing is another instance where the way that you handle your material makes a big difference. So as you can see, they're very carefully rolling and processing this because the speed and the way they handle it makes a big difference in when they're forming this network here, because they want it to be in this kind of amorphous state so that they can shape it to what they want it to be, if it's like a vase or if it's a horse or some sort of unique structure. So making sure that your network is of the correct configuration is very important, not just in bread, but also in other things. Next thing is to rest the dough. So you want it to rise. So you have rising agents that create gas. You also let it rest so that there's kind of like a little bit of give inside your network, because you want to blow it up like you blow a balloon. And so this is an example of what we call an electromicrograph. It's a type of microscope, basically, of when you let your dough rest and then you basically knead it. And so you can see that if you look really, really closely, there are holes inside of your dough. And so when you knead it, for instance, once versus twice, this is the difference. You can get some more regular shapes versus in one versus the other, which is why sometimes they will say knead once or knead twice, because it just leads to a different consistency of your taste. Kind of a matter of preference, right? And so you might think, that's interesting. How is this relevant to other stuff? And the answer is, it's actually pretty relevant to my research too, right? Thinking about how do I make pores? The brain is also a very porous material, as it turns out. And trying to create something like that is very important if we want to study how the brain works. Because as it turns out, putting neurons into just a hard, cold glass plate doesn't always give us all the answers we want if we want to learn about how the brain works. So bread, brain, same thing, right? And now, so finally, you want to process your dough, and so you add heat. What does heat do? It's a physical process. It makes the gas expand. You're blowing up your bread like a balloon. You stiffen the matrix, so you take your net and you cause the gluten to basically lock into place, and it becomes tougher, so it's cooked. That way you don't give your customer raw dough. And there's also chemical reactions. You can ask your local chemist. It makes a tasty crusting. And so how you process your heat matters a lot too. Like I said, processing matters, and here's an example of why.
Okay, this is taking too long. I need to speed up the cooking process. So, if it takes 20 minutes to bake them at 250 degrees, it should only take 2 minutes at 2,500 degrees. Whoops.
Yes. So as you can see, material scientists have to have a good amount of common sense in reminding other people that processing matters a lot. You can accidentally blow up your lab if you don't think about it. So it's not just about adding heat, but it's about how you added it, right? toasting your bread, right, for different amounts of time, different amounts of, for instance, distances from your source of heat gives you different results. And so how is this relevant to, for instance, other materials? And it's actually highly relevant, for instance, in metal processing. This is something that has been known for actually hundreds and hundreds of years by the Japanese when they're making their swords. So the Japanese, the samurai swords are actually curved, but they start off with a straight rod. And what they have to do is that different segments of the sword have to be heated up to a very precise temperature and is held at that temperature for precise amounts of time and processed, hammered into shape at basically very precise ways. And by doing this, they eventually get the sword to become this curved shape where the middle is a lot more flexible so that the sword doesn't break every single time you try to stab someone. And so the edge is very, very sharp. And the way to do that is that you can't just take the whole sword, stick it into an oven, and then take it out, hammer it, and then expect it to be the perfect sword. You have to go and treat every part of the sword differently. And so the way you add heat matters a lot. So, closing up shop, if you've ever seen anyone cook before, you might think that this is all obvious. And the answer is yes, but it might take a long time to figure out. And so metallurgy, biomaterials, crystallography, ceramics, these are all big fields that it takes a long time to figure this stuff out for. And so this is, for instance, one example of, I walked into class one day and was like, what the heck is going on? I thought material science was kind of intuitive. But as I started to sit in class, about 3 months later, I realized that actually, this is just a recipe book. It tells us how much temperature and what kind of stuff we need to put together in order to create, for instance, different types of metal, different types of ceramics, all that kind of stuff. I mean, this is basically multiple people's PhDs worth of work, but these phase diagrams, which initially look very scary and complicated, are just recipe books. And so just to close up why I love material science, I think it's a really cool way of viewing the world. It's a way of approaching problems that sometimes other people might not think about. And it's a way to study anything, which I think that is super duper awesome because I want to study the brain, but I also want to still learn physics and chemistry, and I want to do material stuff. And this is a type of science that allows me to do all of that. And I think that's it, and I'll take questions now.
Please raise your hand if you have a question. I can start with like a first question, if that's okay.
Yep.
I was wondering, so you talk about like determining the recipe and things for the purpose that you want. For example, if you want to make a ceramic, do you do experiments, or like what type of experiments do you do, if any, to determine the recipe?
Like what kind of experiments we do?
Yeah.
So I think it depends on what you're looking for. So for instance, if you're looking for particular types of structures, then what you might do is, if you're looking at, a lot of times, like the way you heat something up, so the temperature at which you bring something up to and the rate at which you bring that temperature, you bring it up to that temperature, you hold it there. After you hold it there, you get some sort of material. I think what a lot of people will do is they take that material and they put it either in like an X-ray machine, they X-ray it to look at the structure and say, this is X material now. Or you might put it underneath a microscope and look at it that way to say what kind of material it is.
Very cool.
So for testing material science for the brain, what experiments do you do?
That's a really, really good question. So here, this is an example of scanning electron microscopy. So it's basically just a really fancy microscope that instead of using kind of optical waves, you'll use electrons instead, which are even smaller than light waves. But some other ways people have done it is that they actually look at what they call like diffusion of ions. So they basically will label different little particles and make them fluorescent, or they'll give them charges, and then they'll stick a little electrode in there and then basically look at the changes in it from like point A to point B and like check the time. Because basically, if you think about the fact that there's a bunch of holes in there, if it has more holes, it'll be able to travel faster from point A to point B than if there are less holes or if there's big holes or there's a lot of obstacles. So that's another way you can like indirectly, for instance, measure it. And then of course there's also a lot of people, there's also ways of just kind of slicing it and then looking at it in terms of either you're making a model of it or you're looking at either samples of human brain, samples of mice models and things like that. But for the most part, it's something that we don't really know a lot about. And we're still kind of developing methods to look at it too.
Can you talk more about the uses of the hydrogel?
The hydrogel? Yeah. So for this particular hydrogel, it was used actually to, so someone else in my lab was making this, and it was used to develop these little microbeads that can then hold neural stem cells. So basically cells that are like little baby neurons. And then you put them into the hydrogel that can keep the neurons nice and safe. And then you can deliver the neurons into a brain that has suffered some sort of injury like stroke. And it keeps the neurons nice and safe until the area of the injury is a little less chaotic and full of immune cells and things like that. And then the neurons kind of will travel out of the hydrogel and then go to the place of injury to kind of grow into new neurons to heal that area. So one way these kind of like hydrogels are useful, A, is that you can use them to encapsulate cells. So that means to basically, it's a holding spot for cells, to deliver cells someplace else. Because if you just deliver cells straight, it's very rough on the cells. So you want to give them something to live in, right? It's kind of like giving someone a car to transport from A to B rather than just making them walk. The other thing is, if you want to just study the brain, it's better to put them in an environment that's closer to what it is like in the body. And this is kind of just a true principle for any kind of biological study. It's just more logistically difficult and complicated. And so just kind of developing this kind of technology is something that we're just starting to do. But that's kind of why it's important to make these kind of hydrogels and things and then start to redo, for instance, some of the experiments we did in the past and see if these same findings hold true from a 2D environment to a 3D environment.
In the recipe books, how did you know how much did he put on them? Like, how did you test it?
This one?
Yeah.
So how is this one tested? I hate to say it, but basically there's a poor PhD student, actually probably multiple poor PhD students, that probably sat and had to combine different amounts of steel and carbon, mix it together, and then bring it up to a certain temperature, hold it there until it melted, and then made some sort of metal, and then take that metal and then test it in some sort of X-ray machine to see what kind of structure they got, as well as like microscopes and stuff, record their findings, and then basically put that as a little dot on the graph. And then you just keep on doing that for every single combination, and voila, you have your map. As you can imagine, that takes a lot of time and a lot of effort. So this one little graph is a lot.
Let's give another round of applause for Cynthia.
All right. So I know it's a long day, but Here we have our third and final speaker, Joy Pajarla, a second-year PhD student in the Department of Physics with a certificate in physical engineering and biology. Joy is interested in studying how patterns and symmetry arise across different living systems and scales. A fun fact is that Joy once led a group of hikers through a mountain range in western Tibet. It's pretty cool. All right, so let's get started.
Hi, everyone. I hope you're having... Can everyone hear me? Yes. Okay, good. I hope you're having a good time. Thank you for staying for my talk. We've had some really wonderful speakers, and I hope to combine some of the topics that they've discussed. So today, I'm going to be talking about the physics in leaves. So before moving forward... This is not working. 1 second, technical difficulties. Oh.
Yeah, it's working now.
Okay, great, thank you. So before I start, let me just give you a little bit of an overview on what I'm going to discuss. So I'm going to give you a little bit of an intro about how I got into science, and then I'm going to talk a little bit about the biology of leaves, and then I'm going to discuss why we're interested in using physics to study leaves, and go into some background, and then go into some of the research that I do. So with that, about me, a little bit about me. So I grew up in Arizona and in India, and I grew up with my family speaking around 4 languages. So I've spent a lot of time trying to understand how to explain things to people in different ways. And so I went to college in Portland, Oregon. It was a really small college. And when I went to college, I really wanted to try and understand the world from as many different perspectives as I could. And so I decided to study physics and some math and some philosophy, and I had the opportunity to do a lot of really interesting types of research. So I studied the neurons and how they develop in the brain and communicate with each other. I studied lasers and what you can use lasers for. I also studied different types of microscopes. And then on top of all of the science that I was doing, I really wanted to understand how we come to know things. So I studied a little bit of knowledge as well. And so with that, I am here getting my physics PhD because I love understanding how patterns work in different systems. And I thought that physics was has a really beautiful way of characterizing patterns across different scales and across different types of living systems. And somewhere in my first year, I found that nature, and specifically plants, have a lot of these sorts of patterns that we can characterize. And so with that, going into plants, you can see a wide variety of leaves here. So can anyone kind of tell me a few of the functions that leaves perform for plants? Yes. Yes, great. Photosynthesis. Anything else? Yes.
They cycle oxygen and carbon dioxide.
Yes, that's great. Okay, great. Those are some great things that leaves do. So all leaves, no matter what they look like, perform the same sorts of functions. So 1, photosynthesis. Photosynthesis is really important for leaves and plants to produce energy. So what that process looks like is carbon dioxide is taken in, and water and light, and then the leaves are able to process that and create oxygen and carbohydrates. So oxygen for us, but carbohydrates for them, so energy for them. Another important thing that leaves do for plants is that plants actually sweat just like us. So when it's really hot outside, the plants are able to, or leaves are able to release water through the bottom layers, through their bottom layers, in order to cool off the plant. And they actually lose about 90% of their water this way. So it's really important. Don't forget to water your plants because they need it to cool off. Next, another important and interesting thing that leaves do is that they defend the plant. And so they are able to fight off infection, predators. And one interesting way that this sort of leaf, it's called the shame plant. The leaves of the shame plant respond to vibrations or touch, and they close up in order to defend the plant from predators. And the last thing that leaves have to do is they provide storage for the plant. So in order to perform all of these different functions and so many other smaller functions, it's really important to store the water, the carbon dioxide, all the things in the networks, the root networks and in the vein networks that are flowing throughout the leaf. And so with that, I've told you all about different types of leaves, and I've also told you a little bit about how all leaves are made of the same stuff. And so when you zoom in, actually, this is kind of a cross-section of what a leaf might look like. And so as you can see, there are multiple different layers. So let's talk about the different layers. So you have the epidermis, which is the skin layer at the top and the bottom. And so the epidermis, as we've learned from some of our fellow scientists before, basically defends the leaf. And so it keeps out all the things that the leaf wouldn't want inside of it, and only lets in, through what's known as the stomata, the things that the leaves need to photosynthesize. And so you have veins as well. Veins are really important for transporting water, oxygen, all sorts of things throughout the plant and the leaves. And the last thing, the really important part of the leaf, this middle layer, is called the mesophyll. And this is where the magic happens. So what happens in the leaf is that sunlight comes in through the top, and then you have this top layer known as the palisade mesophyll, which is responsible for a majority of the photosynthesis. So all these column-type cells basically filter the light, and they're able to process photosynthesis up here, and some of it also happens down here. And then in this spongy area, which is spongy mesophyll, as you can see, there's lots of air pockets in here. This is where the gas exchange happens. So this is where, when someone mentioned all of that processing of water and carbon dioxide and oxygen, this is where all of these things are happening. And so remember again what we talked about in the beginning. We have all of these different sorts of leaves, but they all have the same sorts of layers. So what's actually happening? What's causing these leaves to look really different from each other? Let's focus specifically for my talk on the skin layer, or the epidermis, and then the mesophyll. So what's actually causing these sorts of variations in different leaves are the sorts of environments that they're in. So as you can see, you have a super cloudy and windy environment, and you get a palm leaf. And then you have a snowy environment, and you get something called snow crocus. And then you have more of a springy, temperate environment, you get these sorts of leaves. And then you have really watery environment, you might get something like this, where leaves are able to float. And so on this big scale, this really large scale, you can see that leaves are super diverse. But when you zoom in, they're also equally as diverse. And so on the left, again, you have the environment, and on the right, you have microscope photos of what cross-sections of those leaves might look like. And so let's actually talk about why it might be the case that these leaves look so different from each other. So zooming into the water lily. So as we've talked about before, here is that epidermal layer at the top, and then here's the palisade mesophyll, and then here you can see that the spongy mesophyll has a lot of air pockets. So can anyone tell me why, or maybe some thoughts on why there might be so many air pockets in this sort of watery environment, and why that might be helpful for the leaf? Yeah, go ahead. Yeah, that's great. Yeah, that's one of the main reasons why there's so many air pockets, is that it needs to be able to survive and float in this environment. But there are so many other things that it also does, but that's one of the main reasons. Let's think about something a little bit more complicated. So let's think about the palm leaf. So here you have a really cloudy and windy environment. And as you can see, that same little area, it's so much more dense. And there's so many more cells, and there's very few air pockets. Now, can someone tell me maybe why this might be the case? Yeah, go ahead.
Because it has to stay there so that it doesn't fly away.
Yeah, that's great. Yeah, exactly. And it has to be nice and sturdy in that environment in order to survive. And so these are just some of the things that, some of the adaptations that leaves have in order to be able to, and plants have in order to be able to survive in those environments. But the truth is that there are so many different things that can lead to all of these variations. Those are some examples, but there's not really one rule that in biology that kind of explains why all of this is happening. there are so many different things. So if 200 years from now, the palm leaf might look slightly different, and we want to try and understand why that might be the case, or the water lily might look slightly different. And so in order to do that, we kind of have to zoom in a little bit more and think about how these different layers themselves are like geometrically formed. What's happening when you zoom into those tissues? So with that, let's talk about where the physics comes in. So I've talked about a little bit from the biologist's perspective what leaves and plants do. And so for biologists, they really care about what a leaf does to survive. So like big picture questions. What are the different adaptations they have? What are the functions they're performing? Some of the things that we've talked about. And an example might be, so the main aim of the leaf is to produce energy in order to survive. And one possible answer is that it has to photosynthesize in order to do this. So it has that one function that it does to produce energy. For a physicist, we think about how a leaf survives. So basically, we think about the tools that the leaf needs or the plant needs in order to be able to produce the different structures or things that help it perform its functions. And so what we care about is zooming in so much more towards the cellular level of these different tissues and looking at maybe how the cells are shaped, the sizes of the cells, the materials of the cells, and maybe how even the cells interact with each other. And particularly for my research, I care about the shapes and how they interact with each other. And so with that, let's talk a little bit about my research question, what I care about studying. So we know that all tissues, whether it be our own, whether it be leaves or plants, they all start with single cells, and they grow and develop into multiple cells of different sizes, of different functions, shapes. It's very complicated. And then, eventually, they form tissues. And so I want to try and understand if we're starting with these many different cells of different sizes and how they develop into these different types of tissues. But before we go into that, let's talk a little bit more about the physics of it all. So let's think about this example. Let's think about Newton's laws first, and because they help us explain these shapes and interactions. So what's happening in this nice little video that I've shown you is that the giraffe is sitting in a car, but then the car hits some sort of barrier or wall, and it stops, but then the giraffe doesn't immediately stop right away. It jerks forward, and then it stops. And it stops by the seatbelt, actually. And so what's actually happening here? So if the wall wasn't there, we would say that the car and the giraffe would just probably keep moving forward and nothing would happen. But when the car hits the wall, it stops. But this giraffe kind of has an inclination to keep moving, or it has this resistance to stopping just like the car did because it's independent of the car. But what actually stops the giraffe is the seatbelt. And so this sort of inclination to keep moving until something stops you or changes your direction of motion is what we physicists like to call inertia. And so that is a really powerful and important topic that I have to consider every single day. And so what this looks like is basically if something is at rest or not moving, it's going to stay not moving. And it's not going to start, stop, or change any sort of direction of motion completely on its own. But if something is moving, it will continue to move unless it stops somehow. Okay, next. I'm sure you've all, in some capacity, heard of this law, force equals mass times acceleration. And this is very powerful, very important. And so in this example, you can see that something that's really big is harder to push than something that's more heavy and big is harder to push than something that is small and much lighter. And so what's happening here? So imagine you're pushing something really tough. Like let's say you're pushing on a wall. That wall isn't going to move because you're so much smaller than it, and the effort that you're putting into pushing it is not going to be enough to move it. So let's say you're pushing a water bottle, for example. If you're pushing a water bottle, because you're putting so much more effort in relation to that thing that you're pushing, it's going to move. And so we relate this with this equation. So force, the sort of effort, the amount of effort that you put in, mass, which is the amount of stuff that something has, the matter that something has, and acceleration, which is how fast that thing is able to change its motion. And so one more schematic, basically. So this is a schematic of you putting force on something, and then the mass, and then it's going to move where you're exerting the force. Okay, next, the law of equal and opposite reactions. So imagine a bouncing ball. I'm sure some of you have gotten the bouncing balls from the prize area. And so this is a really powerful example of this sort of law. So what's happening here is that basically when the ball is touching the ground, the ball is exerting a force on the ground, but the ground is also exerting a force back up on the ball. And so it's able to bounce it back up. And there are lots of complicated things happening here, but the most fundamental thing is that if this law wasn't the case, the ball wouldn't necessarily bounce back up. And so this is how physicists are able to equate different forces. And so if one thing is exerting a force on another thing, that thing is also exerting a force back on its own. And so on the original thing. And so this is actually, another example of this is when you're walking. When you're walking, you are pushing the earth back a little bit. And so that's what helps you walk. And if this wasn't the case, you wouldn't be able to walk or move forward in any way. Okay, so with this, let's talk a little bit more about leaf cells. So I've told you all about how physicists want to simplify things and kind of look at them on a smaller level to understand them geometrically. And so with leaf cells, we can actually think of them as squishy interacting objects. And so let's think about a simple case. So let's say you have this box, you have a bunch of particles inside that are squishy, they're moving around, and you look at that box at 1 second. And this is what that box looks like. Let's say later on, you look at the box again. And this time, those squishy objects have actually stuck together. They've glommed together and kind of deformed in that process. So in order to understand what's happening, like I've mentioned before, with shapes and interactions, what we want to calculate from this is the forces. So how are these different things exerting forces on each other? How much effort does it take to be in this sort of configuration? And then perimeters, which is kind of like a proxy or the way that we think about shape. How have the perimeters changed from the previous step? And so you might be able to do this problem just on your own with pencil and paper, no problem, with some equations too. But what if you wanted to think about a more complicated case where there's lots and lots of particles and you want to consider what happens at 10,000 seconds? That would be a really complicated thing to try and do completely on your own, and it would take a really long time. And it would take me a really long time, and I'm getting a PhD in this. And so how do we actually do these sorts of calculations? We actually use supercomputers to do the math for us. So supercomputers are basically like giant calculators. They're able to do all of these different computations that our brain can only do so quickly. So when we put in the physics, they're able to do the math for us, and then we can interpret the results. And so what we want to put in is basically Newton's laws, which we've talked about, how we want the different cells to interact with each other, and how we want their shapes to change. And so when we do that, we're able to get how these different cells actually form tissues. And so, a lot of scientists do experiments, and you might think that that's just, being in the lab and changing different variables and, pouring different liquids and whatnot. But this is also a form of experimentation. So we can change all of these different things and then see what we get out and use that to inform what we do earlier on. And so with that, I've talked a lot about all of the different things that go into leaves. Let's look at some examples. So I've talked about the cell epidermis, I remember, up here and also down here, but we're going to focus on this one. And so this is a picture of the epidermis. As you can see, there's lots of really interesting kind of puzzle-shaped cells. And so what we want to put into the computer is, these are the rules, these are the physics. So we want cells that are very sticky. We want cells where they're growing. And we also want cells, the edges of the cells, to be able to bend really easily. And so these 3 rules are actually able to generate something like this. So as you can see, as the cells are interacting with each other, they're starting to kind of bend and deform in different ways. And this doesn't look perfectly like what we saw before on the left of the epidermis, but it's pretty close. And so this is just a matter of maybe tweaking some different rules and getting something much more similar. So this is what I work on. So let's think about another layer of complication. The boundaries of the tissues actually also affect how they grow. So when you think back to the mesophyll that I was talking about, the middle layer, if you recall, it was surrounded by all of these different types of tissues that are kind of pushing on it and tugging on it. And so we can also model that. So first, we care about growing the tissue, so we can grow the box, but then we can also apply pressure. So when we do this, it's not immediately clear what might happen. But these 2 things working together in conjunction with those previous rules actually produces the mesophyll, the spongy mesophyll in particular, the one layer that has all of those air pockets. And so this is the mesophyll. This is a picture of the mesophyll, and you can see that it has this porous network. And these are the rules that you want to use. So as you can see, the same sorts of rules in general. But here we're adding boundaries growing, and then we're also adding boundary pressures. So when we do that, we get something like this. So as you can see, the cells are growing, they're interacting, but because of these changes in the rules, we start to get all of these different air pockets that are very similar to what we saw before. And so this is the power of physics. We can put in a few rules and get the exact pattern that we want. And so with that, I just wanted to kind of emphasize a little bit more about, what the power of physics and specifically working with supercomputers can do. So here is a video made by someone in my group of what the view that an oxygen molecule might see as it's traveling through that mesophyll area. So all of these different cells that you can see that are different shades of green are actually captured using real experiments. And so this is the real inside of a leaf, and it's really powerful. And so with that, let's go into a little bit of a summary of what we talked about today. So we talked about how environments contribute to diversity of leaf shapes, and how these diverse leaves, even though they are diverse, when you zoom in on the microscale, they look the same, and they have the same sorts of tissues. And we talked about how even the tissues can look different. There could be different amounts of them. There could be different shapes to them. But we talked about ultimately how we can simplify that problem down to just considering the different tissues and how the geometry of them informs their function. And then finally, we talked about how physics is able to look at the geometry and able to look at these cells and kind of determine the sorts of rules that lead to these patterns. And so this is one of the main reasons why I chose physics, is because I love pattern formation, and I love that you can come up with simple rules to identify those patterns. And so with that, here's a picture of my lab group, and I hope you all enjoyed my talk. Any questions?
I heard that leaves, they could feel pain, like plants, they could feel pain. Is that true?
That's a great question. I've also read that article. I am not a biologist, but I can maybe take a guess at that. So I think that leaves and plants have, they communicate with each other through these different networks. And so they can send probably chemical signals to each other or different parts of the leaf about how the plant itself is doing. And so when you kind of disrupt the plant is able to send chemicals to different parts of itself and interact that way.
All right. Do the carnivorous plants also do photosynthesis?
I think they do photosynthesis, so I'm not an expert on carnivorous plants, but I think they also photosynthesize, but I think one of their main functions is being able to break down, insects and whatnot to get nutrients that way. So I think they're complicated.
What is the most unique plant structure that you've encountered over your career?
Oh, that's a great one, actually. I have two, actually. So I recently read about this sort of flower that's able to mimic like flies being on its petals. And so because it's able to mimic flies being on its petals, different flies, male flies, fly to that plant and are able to pollinate it more because it has this interesting mimicry attribute. And so I thought that was super cool that it's able to just do that on its own. There's this other plant, I think, that grows in like the jungles of Amazon that's able to mimic leaves of its surrounding plants. So even though it's one type of plant, it could have up to 3 different types of leaves, which is super cool. Yes?
Thank you for the very nice talk. So I saw that in the last model that you showed to simulate the growth of the leaf cells, you mentioned one of the rules that you applied was that the boundary of the cell parameter, the boundary of the whole cell group would just grow. How do you determine that rate of growth?
Oh, that's a really interesting question. So these simulations are actually not just done, completely on our own. We actually need to get experimental data from collaborators. And so we get that data, and at different stages of a plant's life cycle, it grows at different rates. And so using that sort of evolutionary behavior of the plant over the course of its life cycle, we're able to determine what would be a reasonable length scale or time scale for growth or other sorts of behaviors.
I see. Thank you.
Yeah. Yes, go ahead.
Are you studying a specific plant species or a bunch of them?
Yeah, so basically I study kind of the geometry of different leaves and flowers. But the main plant that we use is called the Arabidopsis thaliana. And it's, I don't think it's a very common plant, but it's very well characterized in biology and botany and stuff. Yes?
Do the simulations that you make change very much when you go from the 2-dimensional pictures to the 3-dimensional picture that you showed?
Yes. So constructing a 3-dimensional, constructing something like this is very complicated. My colleague is very talented, and he was able to put this together. But basically, when you go from 2D to 3D, you not only have to think about how the different edges are touching, but you also have to think about airspace, how tearing could happen, how things could grow lopsided. There's so many things to think about, and so it's considerably more complicated. Yeah, go ahead. Either of you could go first. Do flowers also have leaves?
Do they also have these same cells?
Oh, yeah, that's a great question. So flowers actually, let's go back to the beginning. Let's see. Okay, this is a good example. So flowers, basically, they also have these same structures, so like the epidermis and veins and stuff, but flowers don't have what's known as the palisade mesophyll, and that's because they are not the main source of photosynthesis for the plant. They have a different function for the plant overall. They help the plant attract pollinators, so they have to look a certain way in order to do that, but they don't supply the energy for the plant in the same way. And so they're missing the one layer at the top. And did you have a question too?
I watched this video where, like, people, like, said mean things to a tree, and then they're experimenting if they could grow faster if they heard, like, nice things. Is that true?
Did you ask, is that true?
Yeah.
That's an interesting question. I think that, you should be nice to plants and people and animals. I'm not sure what the relationship would be in terms of saying nice or mean things to trees or other things, but I think that respecting a plant and saying nice things to it, I think, would be beneficial for you. I'm not sure about the plant, though.
I think we'll take one last question. Okay, let's thank the speaker.
I think we have one more question.
Thank you. Sorry, I'll make it quick. This is really fascinating to see this process of observing these plants and then deriving from them the patterns that inform their architecture. How do you see this work being used to contribute to the fields going the other direction, modifying these patterns and formulas to synthesize artificial structures that can be used like plants?
Yeah, that's a really great question. I have not thought that much about it, but I can try and think about it right now. So I think that plants, obviously, and lots of different biological systems are optimized to be able to perform certain tasks. And so one incredible thing about leaves and the sort of different layers, so let's take, for example, the water lily. Oh, let's go back. So let's just look at this as an example, is that these different layers, so specifically the palisade, the cells in the palisade are able to act like kind of microfibers, so the sort of fibers that, or fiber optic cables, so the cables that bring, TV to your TV, and filter light in very specific ways, and these different cells inside act like mirrors. And so they're able to refract the light in different ways throughout the inside and produce just the right amount of energy necessary for the plant to survive. And so this sort of efficiency that you can see, I think I would assume would be very relevant for different sorts of settings where you want to capture lots of light, but you don't have that much energy. So efficiency and usage of the energy budget is really important in living systems, and I think we could also benefit from something like that.
Thank you very much.
Yeah.
All right, let's thank Joy again for...
Sorry about that.
All right, that's it. Thanks, everybody, for coming. Thank you for staying for the talks, and I hope you had a really nice time in today's event.